It's nice to meet everybody. I'm Avery Maddox, MD PhD student in Rao Lab, and today I'll be presenting on high-dimensional weighted gene co-expression analysis for use with spatial data. So what is weighted co-expression network analysis? We'll just sort of start there. So this is working with data that is in this network structure here, where we have our genes as nodes and the co-expression between those genes as our edge weights. And we can take this network and further break it down into clusters of tightly co-expressed genes, which we term modules. And the way we actually get this network structure is we start with the expression matrix. We then calculate this adjacency matrix. So each element representing the adjacency between gene I and J. And that comes from the correlation between those genes that's then raised to a power. And raising that power is a form of like soft thresholding the edge weights. So instead of having like a hard cutoff, you raise it to a certain power to make small values very small and to keep large values, relatively strong. The approach we'll be using for this network is what's termed a signed hybrid. So it's actually a combination of, hence the name hybrid, soft and hard thresholding. So if the correlation is positive, we just raise it to that power like normal. But if its correlation is negative or zero, we just set that edge weight to zero. So we're just focused on these positively correlated genes. We then take that adjacency matrix and compute a topological overlap matrix, which incorporates the similarity between gene I and J, as well as their kind of similarity profile against all the other genes here. So with that topological overlap matrix, that Tom similarity matrix, we then go on to perform the module detection, which starts with hierarchical clustering performed on the Tom similarity matrix, and then followed by a dynamic hybrid tree cut algorithm, which finally gives us our module assignments. So, why do we want to work with data in this form? Kind of what can we do with it that's interesting? This is sort of the little sales pitch for, why is WGCN an interesting thing to do? There are lots of methods to analyze our data, and most of them are designed to analyze features in sort of a one-by-one fashion, which can lead us to oversimplify the underlying biology and miss out on the big picture or the sort of broader context that these things are happening in. So with WGCNA, we can discover interesting pathways, and then we can investigate if these pathways are driven by a certain condition or if they're specific to a certain condition. Further, we can look in these pathways and discover the relationships between our features of interest. So if it's the relationship between genes with each other or the relationship between pathways with each other. And then finally, we can look into our interesting pathways or pathways that we're interested in and discover which features are driving these pathways, which genes are driving these pathways. And we can look at genes that have been identified previously in other studies and place them in a new kind of functional context. So what high-dimensional weighted gene co-expression analysis does is it extends WGCNA to single-cell and spatial transcriptomics data sets. So why can't we just use WGCNA for this? The main issue just comes from the fact that SC and ST data is kind of sparse and noisy, which can lead to spurious correlation measures. And in addition, a lot of the packages like WGCNA itself are not really designed to factor in specific cell types or spatial regions into their analysis. So HDWGCNA addresses these problems specifically by one, it combines highly similar cells or spots into what are termed metacells or metaspots, which helps to reduce that sparsity issue. And then also within the package, it follows a modular design that lets us perform our network analyses within either specific spatial regions or within specific cell types if you're working with SC data. So what does that spot construction look like in HDWGCNA? So you would have a spatial, a cut of tissue that you've done like Visium sequencing on and annotate specific regions within it. And then you can construct your metaspots specifically within the bounds of those regions. So you don't have to worry about, metaspots containing a little bit of two regions if you don't want that to happen. And we start with these principal spots, and it's pretty straightforward. You just gather the spots around it to construct your metaspot, which you can construct by either taking the average or some custom function to then create your metaspot value. And your choice really depends on like what kind of normalizations or transformations you've done prior to then applying your kind of metaspot construction. So now that we've sort of laid out the basics for WGCNA and HDWGCNA, I'll be going through a analysis that we performed using it on glioblastoma. So glioblastoma, it's the most common and lethal primary malignancy of the central nervous system in adults. And one major obstacle to effective treatments is that it has a really high degree of intratumoral heterogeneity. So within a single tumor, we have a lot of different cell states and environments that are or are not susceptible to different treatments or can adapt and drive treatment resistance. Makes it a really complicated thing to target. And this heterogeneity can be broken down in kind of two very broad ways that we did for this study. So one, just from an H&E stain, you can see distinct histopathologic niches. The first would be pseudopalisading around necrosis, which is tumor cells sort of fleeing these hypoxic regions, and they form the pseudopalisading structure here. We also have regions of microvascular proliferation, where we see new vessels forming within the tumor. The highly cellular tumor core, which is just a bunch of these tumor cells, overgrowing within the tumor. And then we also have the leading edge, where our tumor cells are invading the adjacent, normal tissue. In addition to these sort of like histopathological ways of breaking down the glioblastoma tumor, recent work has also helped to identify distinct tumor cell states. So in this work from Neftel et al., they identified 4 distinct tumor cell types, so the OPC-like, NPC-like, AC-like, and MES-like. And they're associated both with different mutations and different kind of environmental niches, which we'll be exploring in this work here. So we gathered a cohort of 44 slides of Visium 10X data for our spatial transcriptomics. This was collected across it's a combination of multiple cohorts, so 14 slides from here at U of M, 17 slides from this Freiburg cohort, and 13 slides from this broad cohort, for a total of 44 IDH wild-type glioblastoma slides. These are taken from tumor resected from donor patients. We then use region markers from the IV glioblastoma Atlas project to score regions and then assign them a label of whether they're CT, MVP, PAN, or ELI. So that's the core tumor, microvascular proliferation, pseudopalisading necrosis, or leading edge, to understand, which distinct histopathological subtype, which microenvironmental niche each of these spots would then fall into. And then we proceed now with those kind of boundaries laid out to go into our network construction. So here we can see that with a power of 4, we achieved a good scale-free topology fit for our adjacency matrix. And the scale-free topology fit is a measure of kind of how close the connectivity is within your network to this kind of ideal biological network connectivity structure. We then take this and use it for our clustering, and we're able to obtain 19 distinct modules through our clustering. So once we have our network, we proceeded with our analysis in kind of three kind of main stages. So first, we look to see if our modules, if their expression was associated with any of these microenvironmental niches. And To perform this, you can use the module eigengene, which is the first principal component of the module gene expression matrix, and it basically captures what the overall expression level is of a given module. To then understand, what these modules actually represent, we can perform over-representations, enrichment analysis using Enricher. And then finally, we can look for drivers of these of modules of interest that we identify in a couple different ways. So one, we can just inspect what are termed module hub genes. These are genes that are like at the core of the module. So they're the genes most connected to the module, which is measured by their correlation with that module eigengene. The second thing we looked at was if there are if ligand receptor co-expression, so if there were specific ligand receptor pairs that were associated with these modules. And then third, we looked to see if there were interesting transcription factors regulating these modules or genes within these modules. So we'll start with our analysis of the core tumor. So this is it's that central tumor mass. It's very highly cellular, and it contains, a bunch of undifferentiated cells. And it's interesting because it's the start of all of the kind of tumor proliferation and specialization that occurs. So we identify many modules that are upregulated in the core tumor, but I'll be focusing on the 4 modules most upregulated in the core tumor for this presentation. And these are interesting. Actually, I'll take a step back. Let me explain this. So on the left, what we have is just the Wilcoxon rank sum testing between our module eigengenes and for the module eigengenes in the core tumor versus all other regions. So it's just sort of a one versus all test of that module eigengene expression. And then again, we, just for the purpose of this analysis, we'll be focusing on those 4 kind of most upregulated modules. And we can get a closer look at, whether these module eigengenes, how their expression relates to specifically to other regions, using a linear mixed effect model of this module eigengene against each region with a random slope and intercept for the slide. And from this, we can see that M13, 14, 15, and 16 are upregulated in core tumor relative to the other 3 regions that we're looking at. So now we can go on after identifying, some modules of interest because they're the most upregulated. We can look to see if, they're enriched for any interesting terms, what these modules represent in terms of pathways. So to start with M13, we see that it's related to glial differentiation, negative neuron differentiation, and vascular transport. Looking at module 14, we see it's related to Notch signaling, glioblastoma stem cell program activation, and interesting, the proneural subtype. For M. And then another interesting thing about M14 is when we look at enrichment for drug targets, we see it's related to, it's a target of 3 PDE inhibitors, enoxamone, milrinone, and amrinone. Looking at module 15, we see it's related a lot to mitosis-related pathways. And interestingly, it's a target of several chemotherapeutic agents, as well as the antioxidant supplement quercetin up here. And then finally, for module 16, kind of confusingly, we see it's enriched for a lot of amyloid beta plaque type pathways. And then just to touch on, we see here at the bottom that it's also a target of vinblastine. So that kind of helps us understand these modules in terms of broad strokes. But to get a more in-depth understanding of them, we can look closely at their module hub genes. So here I've subset the network just to those modules of interest and their top 10 hub genes for each of those modules. So looking at M13, we see several marker genes of the NEFTEL AC state. So that's MLC1, PON2, GATM, EDNRB, TTYH1, and ATP1B2. I won't keep reading out all the gene names for everything, so we have this interesting association of this module 13 with NEFTEL AC state. And we also identify some hypoxia response-related genes. And in terms of, what could potentially target this pathway, we see that PON2 is a target of valproic acid. In module 14, we see a lot of genes related to glioblastoma stem cell proliferation and maintenance. We also identify a lot of genes related to the NEFTEL NPC1 tumor state. So we're beginning to identify distinct modules associated with distinct NEFTEL cell states. And in addition, to kind of give us more context about, the environment that these particular modules might be interacting with or what they kind of depend on. For example, in module 14, we see extracellular matrix-related hub genes BCAN and CSPG5, along with this hypoxia-driven HES6, which could implicate like particular environmental conditions that these, or particular environmental interactions for these modules. For module 15, we see a lot of genes related to promoting proliferation, preventing apoptosis, or both in GBM, as well as, interestingly, two genes, TYMS and HMGB2, that were identified in literature for modulating temozolomide resistance, which is like the standard of care for treating glioblastoma. And finally, M16, which had a lot of those amyloid beta plaque terms enriched in its pathway. We see CST3 and CLU, which are also related to amyloid beta plaque formation. But along with S100B, GFAP, and AQP4 are also markers of the NEFTEL AC tumor state, as well as several other genes that are related to immune modulation and GSC protection, glioblastoma stem cell protection. So that's a lot of details, but what that helps us do is paint a picture for the key genes within these modules. And going kind of through the literature review for each of those, we've begun to connect prior work on these individual genes and now placing them in the context of these modules and pathways into sort of coherent pictures of cell states, environmental interactions, and potential treatment targets, as well as mediators of resistance. So before I dive into the transcription factor regulatory network analysis, I'll just provide some more details on it. So a transcription factor regulatory network, it's a directed network where we have transcription factor and then its action upon its gene targets. We start with identifying candidate TF transcription factor gene pairs using the motive matcher algorithm. And then we can clarify this relationship using the XGBoost algorithm, where we model those candidate TFs against the gene's expression to identify which transcription factors are most important in predicting target gene expression. And we can use that measure of prediction importance to define our regulons, which are comprised of a transcription factor and their strongest targets. So what we end up with is this directed network that's essentially telling us what is the relationship between transcription factors and target gene expression. So zooming in for module 14, we looked at regulons defined by transcription factors that were at the hub of module 14, and we see some interesting relationships emerge. So we can see ASCL1 acting on SOX8, and we can see SOX8 acting on OLIG2, and then we can see OLIG2 going on to extend its interactions elsewhere. So we've kind of expanded just having these correlations from our gene co-expression network into actually understanding how these hub genes are interacting with each other, affecting the expression of each other. to get a more clear picture of what actually might be going on within these modules. And then we can verify if these regulons that we detected actually have an association with the region. So to explain this figure here, each point is representing a transcription factor, a kind of regulon defining transcription factor, and they're colored by their module assignment within the gene co-expression network. The diamonds represent transcription factors that are differentially expressed, and the solid black outline denotes if their regulons are also differentially expressed in relation to this region versus all other regions. And on the X, we see the positive targets of this transcription factor, so the positive regulon. And on the Y-axis, we see the negative targets of that transcription factor, the negative regulon. And what we're looking to see is if the positive targets are increasing with this transcription factor, and then the negative targets should also be then decreasing. That would be kind of the coherent effect we would expect from the data. And we see that here with M14, we identify several of these differentially expressed regulons from our Module 14 hub genes, including SOX8, TCF12, ASCL1, and S6. So then we can also look to see if there are specific ligand receptor pairs that are associated with our modules. And we can do this by looking at the ligand receptor co-expression association with our module eigengenes. So this work is in large part thanks to Reva, who performed the ligand receptor co-expression analysis on a single-cell RNA-seq data set of glioblastoma to obtain cell type-specific ligand receptor interactions, and then map that data onto our spatial data to then look for ligand receptor co-expression associations with our module eigengenes. So one interesting association that emerged from this is these interactions between Col6A3 and LAMC3 with CD44. So we can see these extracellular matrix type interactions with CD44. And based on their co-expression with M14, which we can visualize spatially here, we can understand that there may be a role of these specific interactions along with M14 in promoting the glioblastoma stem cell maintenance in the core tumor. So next, looking at the microvascular proliferation region. Again, we perform a similar differential expression analysis with our module eigengenes and identify three modules, M7, 5, and 17, that here are specifically upregulated in the MVP in relation to all the other three regions. To get a better understanding of what these pathways, what these modules would represent in terms of pathways, we go on to perform our term enrichment analysis, and we can see that M7 is related to angiogenesis and extracellular matrix organization and the epithelial mesenchymal transition. We see that M5 is related to the inflammatory response. And we see that M17 is related to this sort of apoptosis related and TNF-alpha signaling via NF-kappa beta signaling, or NF-kB. And also interestingly, that this specific pathway, M17, might be a target of alitretinoin. So within M7, we identify a couple interesting hub genes, like TAGLN, Notch3, and ITGA1, which have all been identified in prior work for mediating resistance to temozolomide. So again, we're replacing some of these individual genes that have been identified with drug resistance within a kind of specific pathway and regional context. not going to kind of skip over. M5 is just a lot of genes related to worsened prognosis. And M17, we see specifically its NF-kappa beta regulated AP1 complex proteins, FOS, FOSB, and JUNB, which have been identified in prior work for promoting glioblastoma stem cell and tumor cell survival and proliferation. So then we can look specifically within our hub genes of M17 and our transcription factor regulatory network. And again, we see some interesting patterns in how these hub genes relate to each other. We see FOS regulating JUNB. We see JUNB acting on EGR2, and then we see EGR2 extending its effects kind of further throughout our network. And again, when we perform our differential regulon analysis, we can see, in fact, that these M17 hub gene transcription factors, JUNB and KLF6, are differentially expressed, along with hub genes from other differentially expressed modules like TBX2 in module 7. When looking at ligand receptor pairs that are associated with this module, we identify a really strong relationship between this COL4A1, COL4A1, ITGA1, and ITGB1 with module 7. And this kind of integrin signaling was identified specifically acting on endothelial and mural cells here within the MVP. And this helps paint a picture that this integrin signaling is helping to drive the angiogenesis specifically here within the MVP based on its association with M7. So then we look at the pseudopalisading around necrosis region. Here, interestingly, we just found that M4 was the only one uniquely overexpressed in relation to all other 3 regions. Also of interest might be M2, which is our second most overexpressed, but it's not differentially expressed in relation to microvascular proliferation. We find that M4 is related a lot to hypoxic stress response, which isn't surprising given that it's the pseudopalisading necrosis, which is a lot of cells sort of fleeing hypoxia. So that's kind of consistent with what we would expect from that region. But also interestingly, we see that it's a potential target of arsenic trioxide, which has had some evidence in literature for being a potential promising treatment of gliomas and glioblastoma. So just looking at genes that are in the module 4, we identify several markers of the NEFTEL MES2 tumor state. So these are like HILPDA, ADM, NDRG1, ACAP12, SLC2A1, and ANGPTL4. It's a lot of markers of, again, a specific tumor state. And we also identify an interesting set of genes, ADM, ANGPTL4, SERPIN1 and VEGF, and CA12, which in the literature have all been identified as potential targets to overcome temozolomide resistance. And here it's interesting that we're placing them in just kind of a specific region and pathway context to help sort of contextualize those findings. With our regulon analysis, we find that hub genes of module 4, BH-LHE40 and EPAS1 are differentially expressed in the PAN region, along with their targets. And they're kind of out here. here we identify this FN1 CD44 signaling, and from the cell type specific interactions and kind of literature review of this interaction in glioblastoma, we can hypothesize, based on its association with module 2, that this could be driving MES-like tumor state induction, specifically in the pan region, through macrophage tumor signaling. So finally, we'll kind of conclude with our last region, the leading edge. So here we have two upregulated modules, M3 and M19, that are upregulated, and the leading edge in relation to the other three histologic niches. M3 is related mostly to neuron projection development and neurotransmission. And M19 is related to neuron differentiation, the epithelial-mesenchymal transition, and interestingly, it's a target of metformin, which has had some evidence in the literature for being treatment for glioblastoma, or a kind of adjunct therapy for glioblastoma. So looking at M3, we identify several genes associated with the NEFTEL NPC2 tumor state. And in M19, at the core of this hub, we identify a bunch of mitochondrial genes, which at first could suggest contamination, but there's kind of a strong case in the literature that in the context of cancer transcriptomics, these could be a very, kind of meaningful signal. This is supported by literature findings for some of these hub genes like MTATP6, CO3, CYB, CO2, and ND4L, which have all been positively correlated with improved prognosis. And it's, which could have something to do with their role here existing primarily at the leading edge, where we're having kind of less dense infiltration of those tumor cells. Looking at our ligand receptor association with these modules, we identify this interesting role potentially of efferent signaling, which along with M3 could paint a picture of tumor invasion being promoted through neuronal guidance, specifically from the cell type specific information, sorry, that this interaction could be happening between NPC-like and neuron cells. And finally, again, in M3, we find this differentially expressed ARNT2 regulum. So that's kind of the bulk of the analysis of using HDWGCNA for glioblastoma. I did want to kind of conclude with just touching on some other approaches that could also have been applied to this data. I haven't worked with these personally myself, but I was considering them because they have slightly different kind of approaches to how to construct the co-expression network. So a tool like Jodo performs a spatial smoothing before it computes those correlations. And then an approach like Hotspot operates on this idea of local correlations, which are correlations computed between genes that are between cells or spots in the same neighborhood when it's doing the correlation network analysis. So depending on specific considerations that you might have, these could be better fit than HDWGCNA, or offer just slightly different results or insights in HDWGCNA. Yeah, kind of in conclusion, I'd like to just give thanks to Waj, who helped supervise the study and collect all of these samples, as well as my PI, Arvind Rao, and lab members who performed other parts of the analysis on this project, especially Reva and Vish. And a big thanks, of course, to everyone who donated tissue for this analysis. And then just references. And I'm happy to answer any questions from the audience.
Any questions?
I have one question. So, thank you for your nice talk. You have presented us a lot of drug targets for its module, right? So, my question is, the input of that analysis would be the gene list, not the quantitative gene expression. Is that correct?
Yeah, could I be repeated maybe closer to the mic? Sorry, I think I caught most of it, but just not the tail end.
Yeah.
Sure, walk up to the front of the room.
Yeah. So the input of the drug target analysis, the input may be the gene list, right?
Yeah, so you're asking about like what is the initial input for this?
Yeah.
For constructing the matrix? Yeah, so for HDWGCNA, the way we performed it is we did some initial QC filtering. So eliminating spots that looked like they're just blood or that had too few counts. Sorry, I'm clicking all the way back through here. Let's see if I can show it. Yeah, but for us, we worked with just the kind of raw counts, and it sums them up, and then it does kind of all the normalization and transformation on that summed up meta spot. But if you want to do it a different way, like you're doing some kind of deconvolution, or you've transformed it or smoothed it or done some kind of pre-processing to it before, you could use a different kind of aggregation of those meta spots just based on what the nature of the data is.
Thank you.
Really impressive data set and analysis. I'm curious if you could talk a little bit about the batch correction or other sort of technical variation that that maybe that you had to handle when doing this analysis, the technical challenges associated with normalization or batch correction.
Yeah, so for this, we had tried a couple of different approaches of performing like batch correction by either like individual slides or the cohort they came from. I think what I'm presenting here was kind of like the most biologically coherent results we got, which was actually just adding them up, doing the normalization and transformation. And outside of that, not really doing a correction for batch effects. What we see, I don't have the slides to show it, but we see a lot of inter-patient heterogeneity that's just kind of scattered throughout the UMAP, where we can see the spatial regions within each slide cluster, but then each slide is sort of clustering all throughout. And I think that's part of glioblastoma, is that just very large amount of interpatient heterogeneity as well. And something with the actual construction step is that we didn't correct for that. Because to a certain degree, if the kind of heterogeneity within the data isn't so great that it disrupts the kind of co-expression calculation, you can just look at what you get and kind of trust in that. But for the module eigengene correlations with the region, that all goes through the harmonization algorithm, which is then correcting for like slide-specific effects. So the correction kind of happens when looking at the trait associations.
Thank you.
Yeah, could I just ask, in your talk, you talked about key genes as being the ones that you looked at. And what I assume you mean by key genes are the ones that correlate within one of your, correlate highly within one of your modules. What evidence do you have that the non-key genes, of which there must be several, I would guess, are not equally necessary for the operation of a regulon, even though their correlation may not be very high with other genes? Am I making any sense?
Yeah, that's a great question. And that's I think that's something especially, so I can just sort of maybe go to one of these. And so are you asking about, like, for the module, why are we assuming that these like module hub genes are any more important than genes out on the periphery?
Exactly.
Yeah, so that's a really great question. Because again, yeah, all we have here is just the correlation. So we're assuming that because these are the most correlated, based on their correlation with the module eigengene, that there's some implication that among this set of hub genes would be the ones kind of most strongly driving that module's behavior. But that, of course, doesn't always pan out, or the kind of main downstream effect could be the thing that you'd want to target in terms of, like, medical or practical implications. Just the thing that you could actually target would be farther out on the periphery than something here in the core. So that's definitely a limitation. And part of how we try to address that is looking at these regulons, which give us a better idea of actual directed interactions between a transcription factor and target genes. Something we could do as a next step to then help elucidate this even further is very similar to having the transcription factor regulatory network, would just be to have a much broader regulatory network of all the genes where we can have directed relationships. The one shortcoming is just that I think it's debated kind of the reliability of those, just given how much is being computed from the data that you have available in terms of the confidence you can have in those, like, kind of directed edge weights. But yeah, I think it's one of those things where just because a gene isn't at the module hub doesn't mean we could rule it out as, like, insignificant, but it being at the hub is evidence that, this is probably an important gene.
All right, thank you.
Hi, nice talk. I have a question about this transcription network. Is the strength of the edges, is it like calculated as the same for gene co-expression network? Like here we have the negative edges. Does that mean like the transcription factor and the gene are negatively correlated? Are they like is the transcription factor suppressing the expression of the gene?
Yeah, so that's something interesting that these transcription factor regulatory networks add, is that unlike in our co-expression network, which is only positive correlations, which is a step taken for interpretability, depending on how you want to interpret your modules, you could compute modules that include both negative and positive correlations. I think that would be, I think I showed that that's like the unsigned adjacency matrix. But that can get a little tough to interpret when modules, include both. But here we can clearly see both kind of probable, like, suppressor effects on genes, as well as suppressing versus enhancing the expression of these other genes. So yeah, orange is positive association, and then blue is negative. And the way they obtain that is the XGBoost just gives an importance score, but then we contextualize that with the correlation between those TF and its targets.
When calculating the strength of the edges, does the negative correlation and the positive correlation like carry the same weight?
Yeah, I guess the same way. Yeah, so something we look kind of looking at it through this lens is we want to see that if it's acting negatively, that those negative targets are going down with the transcription factor, and that its positive targets are going up. Here you can see, a couple transcription factors will appear off this diagonal, and those are kind of like, I would almost want to say like an almost not like nonsensical effect of that TF then, where its negative targets are up and its positive targets are up, whether or not the transcription factor, and the transcription factor's up. So you would have less faith in those. But in either of these two quadrants here is kind of where that coherent picture emerges, where we see it increasing the expression of its positive targets and decreasing the expression of its negative targets.